Brain cell changes in Alzheimer's disease mapped. Brain cell changes in Alzheimer's disease mapped. You're listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting, presented for World Reading Club, in association with Uniquilibrium. This edition's reading focus comes to us from neurosciencenews.com, and it's titled Brain Cell Changes in Alzheimer's Disease Mapped. It was published on February 2nd, 2023, under the categories of neurology and neuroscience. Summary. A new method dubbed STARmap PLUS was utilized to track and map changes in tau and amyloid beta in the brain as Alzheimer's disease progressed in mouse models. Source: MIT. A common sign of Alzheimer's disease is the excessive buildup of two types of protein in the brain: tangles of tau proteins that accumulate inside cells, and amyloid beta proteins that form plaques outside the cells. Researchers don't know how these protein deposits are related to the other major hallmark of the disease, the death of neurons in the brain. A study by scientists at the Broad Institute of MIT and Harvard, published today in Nature Neuroscience, hints at some answers to this question. Remember, when they say published today, this article was published on February 2nd, 2023. The team used a new method they developed to reveal how brain cells located near these proteins change as the disease progresses in a mouse model of Alzheimer's. The technique, called STARmap PLUS, is the first to simultaneously map gene expression of individual cells and their location, as well as the spatial distribution of specific proteins in intact tissue samples. The researchers used their approach to study brain tissue from the Alzheimer's mouse model at two different stages of the disease and at high spatial resolution. In the earlier stage, they observed a central core of amyloid plaque surrounded by a type of immune cell in the brain called microglia, which are known to play a role in Alzheimer's. The microglia that were closer to the plaques showed genetic signatures that have been linked to neurodegeneration. The scientists also found outer shells of two other types of brain cells that emerged later in the disease. The core shell structure and differences in gene expression of cells surrounding the proteins give scientists a clearer picture of how cells respond to the protein deposits in the brain. Insights that could one day help scientists evaluate existing Alzheimer's treatments and develop new ones. From these kinds of studies, you can infer what's going on in a far more detailed way than you could if you just looked at cells from dispersed tissue samples that don't have their spatial context anymore, said Morgan Shung, co-senior author of the study, a Core Institute member and co-director, Stanley Center for Psychiatric Research at the Broad, and a professor of neuroscience at MIT. This is a new dimension of transcriptomics, and I think it's going to be really impactful. The study builds on a previous version of the technique called STARmap, developed by Xiao Wang, who is a co-senior author on the study, a Core Institute member and Merkin Fellow at the Broad, and a professor of chemistry at MIT. This is an exciting improvement on STARmap because we can now co-map the entire transcriptome together with proteins in the same tissue slices, and many diseases involved changes in protein localization and post-transcriptional modifications, said Wang. The project is also a collaboration with Genentech scientists and was led by co-first authors from the Stanley Center, Hu Zheng, a postdoctoral fellow, and Jiahao Huang, a graduate student, and Haowen Zhou, a visiting researcher. Making a map. To analyze tissue samples using STARmap PLUS, Wang's team used molecular probes to detect specific mRNAs and amplify them as DNA sequences. They also used antibodies to label and identify specific proteins. They then chemically treated the tissue to anchor the DNA and proteins in their native positions within a gel. Finally, they used in situ sequencing and imaging to create a three-dimensional map of the tagged proteins as well as the expression of more than 2,700 genes. The scientists found that processes such as the brain's inflammatory response and the differentiation of glial cells, such as the microglia, were connected to disease progression. Though other researchers had previously observed a core shell structure around plaque, the new gene expression data revealed that the microglia were more activated to trigger an inflammatory response closer to the plaque. The scientists say that this implies that the microglia likely activate near plaques, possibly recruiting other cells to form the outer shells surrounding the plaques, rather than activating far away then moving closer. Understanding when, where, and how microglia activate could be an important part of deciphering their role in the disease. Here we have a first featured image. It looks like a scan or something. It actually looks like an abstract painting, to be honest with you. But I'm sure that's not what it is. Here's the caption, which reads, "Fluorescence microscopy image of brain tissue from a model of Alzheimer's showing tau tangles in red, amyloid plaques white, RNA green, and cell nuclei blue. Credit Zheng H., Huang J., Zhou H., and others." Wang says that a key advantage of STARmap PLUS is that it collects both protein and gene expression information from a single sample, making it easier to align and compare different kinds of data at a high resolution. It can also detect features smaller than cells, which helps distinguish individual cells even when they are densely crowded together in the brain. STARmap PLUS is also scalable and could be adapted to map other proteins or even the entire transcriptome. Beyond Alzheimer's. The researchers say that a crucial next step will be to use the approach to study Alzheimer's progression in human brain tissue samples. This will help determine the extent to which cellular changes that occur in mouse models represent processes in Alzheimer's patients. In animal models, scientists could also use the approach to answer questions about new treatment strategies. For example, if antibodies are able to reach and clear plaques, do the nearby microglia return to their unactivated states and move away from the plaques? Does eliminating plaque or inactivating microglia prevent nearby neurodegeneration? STARmap PLUS could also help researchers understand other diseases, such as cancer, to learn more, for example, about how immune cells attack tumors. The method could also contribute to studies on schizophrenia and other brain disorders. There are mouse models of psychiatry where we know from other studies that there are many different things happening in different parts of the brain, Sheng said. It'll be just gorgeous to be able to see it all in one swoop. funding. This work was supported in part by the Searle Scholars Foundation, the Stanley Center for Psychiatric Research, and the Merkin Institute. About this Alzheimer's disease and brain mapping research news. Author David Cameron. Source MIT. The image I referred to earlier is credited to Zhang H., Huang J., and Zhou H., and others. It comes from the original research, which is under closed access and is titled Integrative in situ mapping of single-cell transcriptional states and tissue histopathology in the mouse model of Alzheimer's disease by Zhang H., Huang J., Zhou H., and others, Nature Neuroscience. We'll be back with an abstract in just a moment. You've been listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting. Presented for World Reading Club in association with Uniquilibrium. Neuroscience News returns with an abstract from the paper titled Integrative in situ mapping of single-cell transcriptional states and tissue histopathology in a mouse model of Alzheimer's disease. Complex diseases are characterized by spatiotemporal, cellular, and molecular changes that may be difficult to comprehensively capture. However, understanding the spatiotemporal dynamics underlying pathology can shed light on disease mechanisms and progression. Here, we introduce STARmap PLUS, a method that combines high-resolution spatial transcriptomics with protein detection in the same tissue section. As proof of principle, we analyzed brain tissues of a mouse model of Alzheimer's disease at 8 and 13 months of age. Our approach provides a comprehensive cellular map of disease progression. It reveals a core shell structure where disease-associated microglia, DAM, closely contact amyloid beta plaques, whereas disease-associated astrocyte-like DAA-like cells and oligodendrocyte precursor cells, OPCs, are enriched in the outer shells surrounding the plaque DAM complex. Hyperphosphorylated tau emerges mainly in excitatory neurons in the CA1 region and correlates with the local enrichment of oligodendrocyte subtypes. The STARmap PLUS method bridges single-cell gene expression profiles with tissue histopathology at subcellular resolution, providing a tool to pinpoint the molecular and cellular changes underlying pathology. You've been listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting, presented for World Reading Club in association with Uniquilibrium. This edition's reading focus has come to us from neurosciencenews.com and is titled Brain Cell Changes in Alzheimer's Disease Mapped. It was published on February 2nd, 2023. You can read this article and others like it for yourself by visiting neurosciencenews.com. That's neurosciencenews.com. neurosciencenews.com. Brain cell changes in Alzheimer's disease mapped.